Hello, everyone, and welcome to today's webinar, End-to-End Tools and Services for Interrogation of CRISPR-Cas Associated Genotoxicity. This webinar is part of the CRISPR Virtual Event Series 2024, brought to you by LabRoots and is sponsored by IDT. For more information on our sponsor, click the IDT logo found on your screen or visit idtdna.com. I am Israel Gonzalez of LabRoots, and I'll be your moderator for today's webinar. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by both communicating with other attendees using the group chat feature and also by submitting questions during the presentation. Now let's get started. I'd like to introduce our speaker, Ashley Jacobi, Director of Applications and Market Development at IDT. You can view our speaker's complete biography below. Ashley, you may now begin your presentation.
Yeah, thanks. And I'm excited for the opportunity to present on some of the new CRISPR innovations that we've been working on at IDT. I'll be presenting on a lot of new developments that are not commercially available yet, but please reach out to me directly if you want early access to any of these, or if there's anything you can't find directly on our website. I'll have my email at the end of this presentation, and happy to directly get in contact with you and get you to the right people if you want some things in here that you can't find directly off of our website. All right. So before I dig into some of our newer innovations, I really wanted to take a moment and just highlight the breadth of the development work that we've done at IDT to enable labs to more efficiently run CRISPR-based experiments, ultimately leading to enabling products to develop therapeutics. So here's just a timeline of a lot of the work IDT has done over the last almost 40 years. And you can see here that IDT has been manufacturing oligos since 1987. And so we know the company has been built upon manufacturing DNA and RNA oligos. And so then when the first CRISPR papers came out, it was really, really exciting times for scientists at IDT. And we really dove deep into how can we to make this exciting technology, much more things like our high fidelity Cas9, things like our different nucleases, or just optimized guide RNAs. And so in 2021, we did launch our CRISPR analysis off-target system, RAMP-seq, and you can see that highlighted there. And this is a tool for people to use in their own labs. So where we provide the master mix, we do all the design of the primers, we provide access to a user interface, again, for scientists to be able to fully do all of these experiments in their own labs with access to IDT's technical support as well. But with the most recent launch that you can see in 2023, where IDT is now manufacturing GMP guide RNAs compliant with ICH-Q7, we have now moved to taking some of these safety technologies and actually offering them as services. We've just had a lot of our loyal customers and collaborators ask if this is something that we can do directly as a service. And so we've listened, and I'm excited to share today some of the various things that you can have IDT directly do in our labs for you. And so as I move on to the next slide, before I get into the technology, I just wanted to take a moment to highlight some of the publications over the last 5 to 8 years where IDT scientists have collaborated with scientists in the field to actually do a lot of these off-target safety assays for them. As I mentioned, we've been developing these and optimizing on, existing technologies to do these safety profiles on our own technologies and our own product development. But alternatively, we've also supported a lot of key opinion leaders in the field and doing the safety work for them. And so this just highlights a suite of manuscripts where IDT has done a lot of the off-target work for various players in the field. And so, I say that because this is not something that's new to IDT. It's just new to us actually offering it as a service offering. And in part for us to be able to offer this as a service, we have worked really closely with SecurityX to take out the license for the foundational technology for the off-target nomination assay we use, GuideSeq. And so GuideSeq, I'll go into a lot more detail of this. IDT's done some work to optimize GuideSeq and incorporate some of our own technologies into this. And now we can offer this as a service for our customers. And so this just highlights that, back in July, we worked with SecurityX, and IDT is now a provider of this technology. So here kind of highlights, before I go into a little bit more detail of all of these technologies, kind of the workflow now you can think about coming to IDT for your safety assessment package. And so this is incredibly flexible and modular, and allows you to really like hone in what you need with our scientists based on the application and where you are in your clinical journey. And so, if I start in the far left, I'm highlighting various components here where we are able to start with doing the off-target nomination. So the way we can do this at IDT is if you have a couple guide RNAs of interest or your lead therapeutic candidate, we start by getting on a quick intake call with you, talking with our scientists and discussing the type of work that you need done. And so we would then send you protocols and our double-stranded tag, which you would then integrate and deliver with your guide RNA and your Cas of interest. And so IDT has optimized the tag that's used for GuideSeq quite a bit. So we would send our specific one, and then you would send us back cell pellets. Alternatively, we can have a discussion around using some of IDT's model cell systems to do the actual transfection. And so what I mean by this is we have cell lines that stably express Cas9, such as an HEK293 stable cell line, where we can deliver the guide RNA, have it manufactured at IDT, and deliver that guide RNA directly into these cells with this tag and do all of that initial transfection piece at IDT as well. And so the figure in the very bottom, what that's highlighting is when we have delivered a guide RNA of interest into cells that stably express Cas9, or delivering the guide RNA as an RMP into 4 different cell types here, which are shown in order from left to right of U2OS, K562, iPSCs, and primary T cells, that when we do those latter cell lines, we are actually still detecting every single cell, every single site in these cell lines that you would have picked up with that Cas9 cell line. We also incorporate a lot of positive controls and things like that. So this really allows us to overrepresent any potential off-target risks in this model system. And then we follow up in some later stages here in the off-target validation, where we would then move back into your cells of interest. So we can go both ways. So to kind of like summarize that, we can send you our tag and a protocol, and then you deliver that tag into your cells of interest with your guide RNA and your Cas nuclease, and send IDT back your cell pellet. Alternatively, you just tell us the guide RNAs that you want to look at, and we deliver them into cells that we have at IDT. So then the next pillar here, the off-target nomination library prep. So then once we receive that cell pellet, or we isolate the genomic DNA from the cells at IDT, we proceed and do the library prep with this GuideSeq optimized assay. And so this is just high level here, where we do all the library prep and the sequencing at IDT. We also have a discussion if you want to incorporate any in silico predicted off-target sites. And then we meet, and in the third pillar here, we have a very detailed discussion of the report. So we put a report together that is, has all of the information that the regulatory bodies are asking for. We nominate all of the sites and present these to you in a tiering approach based on various characteristics, things like how reproducible they are across biological replicates, where they hit in the genome, and various other tiering strategies that we employ. We then get you this report, as well as an Excel supplementary data set that has all of the raw data, access to your FASTQ file, all the metadata of everything that's nominated. And then also in the report, we have a lot of graphics and figures that you can use directly in your IND filings as needed as well. So then following that, we can stop there. We've done the off-target nomination for you, or we can move on to also doing off-target validation or confirmation for you. Now, where we are going to take this nominated list of sites that we have agreed upon together, and we're going to make a multiplex amplicon sequencing panel, which we use at IDT as RAMP-seq, and we're going to do deep sequencing in your cells of interest at around every genomic loci that we have nominated, and actually really confirm if there is a level of editing down to 0.1% that we're able to detect. And so when we get to this stage, we have aligned on the list of sites that we want to interrogate. You would send us additional cell pellets now that don't have this tag in it. And then IDT would do the design of the sequencing primers. We would do the library prep, and then we would do the sequencing and the analysis. As I mentioned at the beginning of this presentation, the RAMP-seq technology is a system that is available to do in your own lab. So alternatively, we can work with you to do that as well. But if you would like to just send all of your samples to us, we can do all of that here. Additionally, we have built a couple analysis features that are not part of the user interface that we have for our RAMP-seq product, where we are able to look at translocations and then do some additional statistical classification. So that comes with this service as well if we do the RAMP-seq for you. And I'll go into a little bit more detail about those. And then lastly, you can see on the right, then we go through a very similar full consultative review of the data where we're able to get this in a report for you. And then that figure in the bottom is highlighting where we're able to do a lot of statistical classifications of any of the sites that we are detecting that have indels, if they're true off targets or not, based on background and other emerging strategies. Okay, so now I'm going to dive into the technologies a little bit more. I just kind of went through the full workflow of what we include in the safety package for you. So again, the first step would be the off-target nomination for the guide RNA and the CRISPR system of interest that you're working with. And so, we've spent several years improving upon the empirical cell-based assays, but at a high level, as I mentioned, we have you deliver the tag and a guide RNA into cells that express Cas9 or alongside your nuclease. And then this tag is then integrated anywhere where there's a double-stranded break. And then when we do library prep, we're able to use that tag as an anchor to sequence off of. And then also what we've incorporated into the IDT version of this is we have primers that are really optimized for efficient PCR and multiplex PCR, the ramp-based primers that allow us to do this library prep in one reaction. So you're no longer having to do a top and bottom strand as you do with the original guide-seq technology, and just get a lot more accurate amplification here. So here's just highlighting some data of this technology that we've developed. On the far left is just comparing if we used the kind of the out-of-the-box guide-seq technology and compared that to IDT's improved nomination technology that we're overall calling and nominating more sites. But in the middle panel, what this is showing is if we take like an apples to apples comparison of sites that were nominated by guide-seq or by the improved IDT strategy, we are nominating them at a same amount. So we're getting very similar UMI read counts there. So we've not changed the quantification there at all. And then on the far right, what we wanted to look at is, okay, now of the sites that IDT is calling, are we missing any of those sites that guide-seq was calling? And what this shows is for this set of data here, we were able to call, it was 99.8% of the sites that GuideSeq called. So we're not missing any of those sites. We're calling more sites, but we're calling them at, the similar levels as the original GuideSeq. And this is just, we've, built this to be a lot more amenable to high throughput and able to incorporate more replicates and look at, many guide RNAs as needed. So then, as I mentioned, after we do the library prep and we do the sequencing, then the next step is to get you a very detailed report. And so here's just a couple screen grabs of the pages in the report where we show all the QC metrics, a lot of details of the sequencing run is in that first panel. We do everything in biological triplicate. We have a lot of data supporting why that's valuable and gives you the least likelihood to miss any sites. And so then that plays into account for the tiering strategy and what we suggest taking into off-target confirmation. So we have a Venn diagram there that highlights the overlap of the various replicates. And then, the third panel there in that blue box is highlighting then, okay, here we go and characterize all the off-target sites that are nominated, highlight the Levenshtein distance, highlight what gene this is in, do a lot of annotations, give you the tiering score, put a MAR risk score in there. And so this is all stuff then that we talk through with you of, okay, which of these sites are things we want to now look at deeper with all of our relevant samples at the clinically relevant doses, and then do the off-target confirmation. And then the data from this off-target nomination report can then directly integrate, it shows on the right, into IDT's RAMP-seq design tool. And so this, again, is our multiplex amplicon sequencing design tool that allows you to design primers around your on-target locus, as well as hundreds of thousands of off-target sites, and get these in one library prep or a couple library prep reactions. So that all interacts together. And then we move into doing the RAMP-seq confirmation so that we can then quantify the amount of on and off-target editing with doing deep sequencing at the on-target site, as well as all these off-target sites. So as I mentioned, this is something that's commercially available to buy as a kit and do on your own, or you can directly send the cell pellet to IDT, and we can do this as a service for you now. So the way our system works is we design primers around all of those nominated sites. All of the forward primers are combined together, pulled together, and all the reverse primers are pulled together. And it's a two-step PCR reaction where the first PCR is where you're doing that locus-specific amplification, followed by a simple dilution into then doing PCR for Illumina sequencing. And then direct sequencing on an Illumina platform. And then that FASTQ file from the Illumina platform can directly be dropped into our user interface that we've partnered with Illumina to host in the cloud. And that's called our RAMP-seq CRISPR analysis tool. This was published in 2021, I believe. And so all the details for that are highlighted in the publication on the right. And this is just a figure out of that publication where the analysis tool that we've built for this is really calling very accurate quantification of the editing. And what we're showing here is this is a synthetic data set of 603 samples where we know true editing is 50%. You can just see the various ranges with using different analytical pipelines. And then all the way at the far right of each of those plots, the CRISP alterations, that's our RAMP-seq analysis tool, how accurate all 600 of those sites were at being called at the correct level, both when we were using a Cas9 or a Cas12a based cutting strategy. So again, this is all available through a cloud and through reagents, or send us your samples and we can just do all of this for you. If you want to see what the analysis tool looks like, we can always get you access to that as well. Or you directly use this. And as I said, it is pretty straightforward, where it's a drag and drop of your FASTQ files from your Illumina sequencer into this upload feature on the left. You, in the middle here, apply some data into a sample sheet, and then it's a pretty quick run of the data. And then you get a lot of graphical outputs, which are on the right, that highlight the levels of editing If you're doing HDR, we can look at the levels of HDR, of perfect HDR. And then this also interacts with IgV and can get you a lot of allelic frequency data. But as I mentioned, we have also built some additional analysis tools over the last couple of years that are not in our online user interface. And one of those things is a way to look at translocations now. Using the same RAMP-seq multiplex panels, where we've built some new analysis features that allow you to, that will take a look at various forward and reverse pairings of different forward primers and reverse primers. And if there's any reads that are called there, to be able to discern if there's any translocations at those predicted sites. And so this is something that we have a Dockerized tool for that will actually incorporate into any of the service offerings we do. And so we refer to this translocation analysis as PASTA, where it's a primer-anchored statistical translocation analysis. And there's a figure representing that there through a collaboration that we did with AstraZeneca, actually. And then, as I said, we just need to take our read, run, and read true FASTQ files and our amplicon bed information, and we're able to run this through a tool and then get you that summarized translocation analysis. What I'm showing on the bottom right is as part of validation of this tool, we took a data set from a publication that's highlighted there, and we're able to quantify the various translocations that were part of that data set at a very accurate level. And so I believe this, is showing around 10% down to 0.3% various translocation levels, where we're able to detect the level of those with this new tool and actually have a very tight background as well when we've looked at these spike in translocations. So again, this is something that anytime that we'll do a RAMP-seq service for you, that we would add on this additional data set. And then kind of as a case study, and just to introduce another product that we have developed and will be commercializing soon, we originally built this translocation analysis when we were developing a new HDR enhancer protein. So this HDR enhancer protein we refer to as HEP, but it is a 53BP1 inhibitor. And IDT has done a lot of work after some of the original publications for this came out to optimize and do some mutagenesis on this protein to increase the binding affinity for that 53BP1, which in turn increases the HDR rates. But as part of doing that, as I mentioned at the beginning, developing products that remain safe are incredibly important to us. So we wanted to not only look at if this HDR enhancer protein had any change to the off-target profile of these guides when delivered with the peptide, but also did they affect any translocations. So that's where we were able to take this existing RAMP-seq data where we're looking at off-targets, do this additional analysis, and look for this at the sites that we had nominated for our off-target risk. So what we're looking at here on the far left, first is just showing the boosts in HDR you get with this protein, and then also looking at a small molecule that we refer to as V2 that we also have available. So on the far left, where it says no enhancer, here we're looking at an EMX1 locus, which we know has a lot of off-target editing. We chose a site intentionally that had high off target to be able to look and see if we were making any changes. But you can see in this case, if we are inserting a single-stranded oligo to incorporate, in this case, an EcoR1 site, so knocking in 6 bases here with Cas9 RNP into, in this case, HEK293 cells, the level of HDR we're seeing of those of that EcoR1 site, it's about 20%. When we include that peptide enhancer, which we originally referred to as UBVA, so UBVA, HDR enhancer protein, same thing, we are able to get this to boost by about 1.5-fold. When we use that small molecule, this does boost it quite a bit higher. But there, depending on the cell type you're in, there can be some toxicity that can come with that small molecule as well. And then in the far right of that same graph on the left, it shows if you use these two HDR enhancers together, you can even get another additive effect. So we have definitely seen advantages where you can titrate down that V2 small molecule and include the peptide. or just use the peptide, which has been shown to be an incredibly safe tool to increase HDR rates. We have to date not seen any toxicity at all in any cell type we've tried. And this is pretty agnostic to CRISPR systems as well as the donor template type. But again, the focus of this presentation is really on genotoxicity and safety. So we wanted to look, okay, in developing this peptide, are we changing the off-target profile at all? So using our RAMP-seq multiplex amplicon sequencing, this is showing the top 4 off-target, the top 4 sites that have the highest off-target editing, now looking at those same 4 conditions. So what we're seeing in the first panel that says OT1 are the total levels of off-target editing that you're seeing at this site. So without any sort of enhancer, at this locus, we get, a little over 50% of reads at this off-target site. When we add in the peptide, that doesn't change at all. However, when we add that small molecule, the rates of off-target at that site did actually increase. And then with the synergistic, the V2 as well as the peptide, it's the same as with the V2. So it's that same data set now, just at 3 other genomic loci at what we call OT2, OT3, and OT4. And what you can see is that peptide is not changing above not having any additive, changing the off-target profile at all. But we do see some increase when we're using the small molecule. So this is just strictly looking at an increase or change in indels at those off-target sites. But what I mentioned that we built was we wanted to also look at translocations. So we took that new tool that we refer to as PASTA, and we are now able to look at the same 4 conditions. So no enhancer, just our peptide, the small molecule, or the small molecule plus the peptide. And we are taking that same set of RAMP-seq data, and we are able to now calculate the percent translocations by summing all the reads using the forward amplicon primer for the site that paired with a reverse primer from a different site, though only for translocation events that reach a 0.05 FDR, divided by all the reads that include the forward primer. So that's kind of a high level of how that's done. But what you can see on the left is that the level of translocations that we're detecting at the on-target site first for EMX1. So, background levels of that without any additive. And when we include the peptide enhancer, the UBBA, that does not change at all. So we're not inducing any translocations. However, with the small molecule, we were able to detect an increase in translocations that were seen at that on-target site, and then a little less. But when you have both of them, that small molecule still has that effect. And then now it's the same set on the right, but instead of looking at the on-target site, we are looking at the same 4 top off-target sites. And what you can see there is also at those 4 locations, we are not inducing any translocations by adding on that small molecule peptide. So this peptide is something that we are now will be commercializing early next year. We're actually working with our sister operating company, Aldevron, to produce this, where we'll have this available as a research grade protein, as off the shelf, as well as a GMP protein. We're seeing a lot of value in this being a clinical tool as well. And things like this is allowing you to reduce the MOI of AAV when delivering AAV as the donor template. And again, like I said, there's not been any sign of toxicity at all when using this in any of the systems that we've tested this in. So excited to have this available. If you want early access to this, like I said, reach out to me, but this will be available next year as both research and GMP protein. Okay, so now going back into the service side of things, we have here the report that you would see if you had us do RAMP-seq analysis for you. So this is, again, just a couple of pages of that deep sequencing report, but we would have the on-target site as well as all the off-target sites and show you the level of editing seen and if there's any translocations. So we incorporate that new PASTA tool, which is in the screenshot on the left, where we're able to look for any translocations. And then on the right, we're able to now incorporate this new off-target classification that we've built. And so what this is for off-target off-target confirmation, we take an approach using in-house algorithms to take indels that are already identified from our RAMP-seq tool and merge them based on different positional features, and use the counts of those merged events for improved statistical testing. So again, this is new and something that would come with the service package, and it's not part of our online CRISPR analysis tool that will just give you the raw indels. But using this approach, we achieve improved sensitivity and specificity on a data set containing a ground truth compared to classical statistical methods like hypergeometric test or a t-test based on indel frequencies. But also as compared to other advanced approaches like those available in the software package like CRISPR, as can be seen in the graphics on the bottom right here. So all of this information gets functionally converted into findings in our report. And so like you can see on the left there, where we'll then be able to report any significant indels we see, the read depth we sequence to, and then additional meta information about sites that we observe to be edited, and then get this, plot of whether any of these nominated sites, now that we've done deep sequencing and confirmation, are, truly having any editing. And then additionally, we include that, like I said, that information regarding any of the translocations that we observed in using the same RAMP-seq data set to produce the indel classification findings. So really a complete package there to get you the off-target confirmation. So I did mention at the beginning that IDT is now manufacturing GMP grade guide RNAs. And that's really what pushed us to build out these tools that we've used for our own product development, or that we've made off the shelf, to have them as a service. We have just really seen an advantage of people now able to come to us and get this service done and then move directly into manufacturing their engineering or GMP guide RNAs. So just a little bit about this before I end. We have a facility now that's up and running in Coralville, Iowa. It's a 41,000 square foot facility. You can see a picture of it here, that manufactures DNA and RNA oligos compliant with ICH Q7 and ISO 8 clean rooms. And the RNA oligos are up to 150 nucleotides in length. And then dedicated labs here with single pass airflow with positive differential pressure in the various clean rooms to reduce any risk of cross-contamination. We have dedicated analytical labs to do all the product testing on site, and then the continuous monitoring of the various environmental conditions. So really excited that we are now able to support our customers who we do a lot of early work in the discovery phase with, now all the way through their clinical programs. And I just wanted to take time to highlight, one aspect of the analytical package that we have in looking at purity. Another Danaher company, SciEx, we have partnered with to use their single channel capillary electrophoresis method using their PA800 Plus system to look at oligo purity. So this is an assay we run at IDT for every GMP oligo that we manufacture, but this just highlights the system and the kit and the separation that comes with the solution from SciEx. But what I want to highlight here is just some of the development work we did to show the N-1 resolution using this technology. And the way we did this was we had 100 nucleotide guide RNA manufactured. And in this case, these were not GMP oligos. These were just research-based oligos when we were developing this platform. But what we show here as N is just 100 nucleotide Cas9 guide RNA. And then we made a 99 mer. And then we mixed ratios of 100% of the 100 mer and 0% of that 99 mer. And then as you can see in that table right there, increasing amounts of that 99 mer. So 90% of the mix was the 100 mer and 10% was the 99 mer, all the way down to where we just had 100% of that 99 mer. And then what you can see in the middle panel are the electrophoresis chromatograms of these various mixes, where you can see the two peaks, where you're able to see the 99 mer and the 100 mer. And then the quantification in this case of the 100 mer is shown there as a percentage. And then off to the right, we have now gone and taken the actual ratios from that table where we know what amounts we put in, and what we're able to calculate is the area under the curve for that 100 mer, and you can see we get an R squared of greater than 0.99. So just getting very good resolution of these N-1 products. And so then here's just a couple of views of some oligos now that were made in that therapeutic facility and doing the purity assessment with this single channel capillary electrophoresis, where we're able to show the full length product of these in the 90% range. In this case, this is 2 different 100 mer sequences that we manufactured in our new therapeutics facility. And then the last thing I wanted to show is we're able to do a lot of additional services and tests for our therapeutics clients as well. And this is doing some functional testing on some therapeutic guide RNAs, and with a lot of different grades of therapeutic guide RNAs, meaning manufactured in our therapeutics facility. So what I'm showing, if you look at the left figure here where it says cell-based editing activity, what we have done here is taken 6 different 100 mer guide RNAs targeting the same locus and delivered them into Cas9 cells as an RNP, into K562 cells with a Cas9 RNP. And then what we have here is 2 different 100 mers made in our therapeutics facility, 2 different lyophilization events from that synthesis event, and then 2 different synthesis events, so the green and the blue. And then we have a purified research oligo in gray, and then in the darkest gray black is just a standard DSAL research screening guide RNA. And so these were delivered in a dose response of 4 micromolar down to 0.1 micromolar. And you can just see the high reproducibility of the green and the blue. So sequences that were made in our therapeutics facility, giving you the highest level of editing at the lowest doses, just these having the most full length product, and just how reproducible that is as well. And you see a little drop in activity with that HPLC purified RUO guide RNA, not as much full length product there, and then a little bit further drop as well with the standard DSAL, which has no purification done on it. So this is just highlighting the reproducibility, the robustness of these oligos made in our therapeutics facility, but also that we're able to incorporate functional testing as a service. If you need any of this done, if you want this type of data for any sort of comparability testing, please reach out. And then the figure I talked about on the left was putting this into cells. We can also do an in vitro assay where we synthesize a double-stranded fragment as the substrate and then do in vitro cleavage. And it's the same set of oligos shown here. So with that, I want to thank the team of scientists at IDT here shown on the left, which are part of the CRISPR product development team that have done a lot of the development of these off-target assays and this HDR enhancer protein, as well as our other Danaher companies, Aldevron, which is now helping us manufacture that HDR enhancer protein, and SciEx, who we worked really closely with to develop the CE method. And then lastly, some collaborators at AstraZeneca that we worked with to develop that translocation assay. And my email, ajacobi@idtdna.com, is listed there. If you have any questions, you can directly reach out to me. Or if you want to have a consultation of the off-target analysis package that we're able to offer, there's a link there that you can go directly to and fill out a couple questions, and then you'll have the team of scientists reach out to you and set up a call where we can talk through doing a project for you. So with that, thank you for your time. I really appreciate getting to share some of these new developments that we have going on at IDT with you, and I will take any questions.
Awesome. Ashley, thank you so much for that informative presentation. Let's go ahead and dive into the Q&A segment of the webinar now. And as we do, I want to encourage our audience to continue submitting your questions for our speaker today. And it looks like we have a few coming in. So let's start with this first one. So Ashley, what is the advantage of incorporating RH-AMP PCR into the GuideSeq assay?
Oh yeah, okay. So good question. So I didn't talk about this too much, but the ramp-seq technology is built off of the ramp technology, which utilizes RNase H2. And so what that does is our primers that we use have an RNA base near the 3' end of them and a blocking group. And so then the master mix that we use for that first round of PCR has an RNase H2 in that master mix. And so then the way that works is RNase H2 recognizes a DNA RNA heteroduplex. So now that we have that RNA base in that primer, when it anneals to its genomic locus, that RNase H2 will recognize that and cleave 5' of that RNA base and release that blocking group from the primer and allow PCR to happen. So that's how multiplex PCR works with this system and why we're able to do hundreds of thousands of off-target assays for off-target confirmation. And so to the question of why did we incorporate that into the GuideSeq, that allows us, that's just a two assay multiplex, but it allows us to get really efficient PCR of the top and bottom strand needed for GuideSeq in just one reaction. So we're not now having to do separate PCR reactions for every guide RNA.
Wonderful. Thank you for that.
Mm-hmm.
Our next question is, what cell types does the HDR enhancer all work in?
Yeah. Okay. So I alluded to this, but I did not actually show any data. But we have now, at this point, tested the HDR enhancer in quite a suite of cell systems that this has all worked in. So we have seen this be successful in iPSCs, in HSPCs, in primary T cells, and then your standard immortalized Cas9s or your standard immortalized cancer cell lines. And also, I think I mentioned this, but it's also pretty agnostic to the CRISPR system. So this has worked with Cas9 nucleases, it's worked with mRNA, it's worked with MAD7, with Cas12a, and then it's also very agnostic to the donor template type. So whether it's a short oligo, whether it's a plasmid, whether it's a nanoplasmid, whether it's AAV. So it really works across any type of HDR application.
Understood. Thank you so much.
Yeah.
We have time for just a few more here. Do you suggest any other orthogonal off-target nomination assays?
Yeah, so the nomination assay I talked about today was GuideSeq. So that's a cell-based assay where you know how I explained that we introduce that tag into your cells of interest or to a model cell system. There are also a lot of in vitro assays as well. And so as I mentioned, we are working really closely with SecureDX, who has the OneSeq biochemical assay. So I see a lot of advantage in doing both of those, where you'd use the OneSeq biochemical assay as well as in-cell GuideSeq, so you get two orthogonal nomination assays, and they'll work together to put the nominations from those two assays into your off-target confirmation. And so that's something, again, as I mentioned, we work really closely with Secure that we can help support getting both of those assays together into a ramp-seq panel.
Great, thank you for that.
Yeah.
It looks like we have time for one more question here. What in silico predictive tool does IDT use?
Yeah, so I highlighted on that first slide where it was an overview of the services we offer in silico prediction. So you can come to us with your preferred in silico prediction tool, or you can utilize the one that IDT has built. And so we've built our own based on our own data sets. But our service is agnostic to using any of the available tools, and there's a lot out there. But we have our own that we've developed.
Perfect. Thank you so much for that wonderful Q&A, Ashley, and also to our audience for submitting questions. I appreciate your time and your important research. And any questions that we didn't have time for today, including those submitted during the on-demand period, will be addressed by our speaker via the contact information that you registered with. This webcast can be viewed on demand for 1 year until October 23rd, 2025, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thank you to our audience for joining us today, and again to IDT for sponsoring this webinar. Next time, thank you.